Okay, so welcome to this next video in which we are discussing transcription factors. Okay, right, so in the previous video what we did was we looked at an explanation for why transcription factors generally bind to the major groove of DNA rather than the minor groove of DNA. What we now want to do is, armed with this understanding, we want to now explore some of the common motifs found within transcription factors or gene regulatory proteins, if you're being more correct. Okay, so motifs of transcription factors. Okay, right. So the first motif that you find in a huge number of different transcription factors is what's known as the Helix-Turn-Helix motif. Okay, and what I should say before we actually even start this is we're going to see a huge number of different motifs. First, I should probably tell you what a motif actually is. So a motif just means a portion of a protein. Okay, so proteins are huge, great things. A little portion of that is what you would call a motif. Okay, so transcription factors are proteins, so we're talking about the motifs of these proteins. So what I would like to start off with is we're going to see lots of these little motifs that are found in many different transcription factors, but many transcription factors will contain multiple of these motifs. Okay? So that's something to bear in mind as we go through this. So the first motif that we're going to talk about, which is capable of binding to DNA and which is found in a great many different transcription factors, is what's known as the Helix-Turn-Helix motif. Okay, right. So basically this motif, or this domain, another word for motif is domain. This domain consists of two alpha helices, okay, with a turn in between them. So let me firstly just remind you of what an alpha helix is. Okay, so basically an alpha helical structure is a secondary protein structure. Okay, and it's where the polypeptide assembles into a spring-like structure. So this line is going to represent the polypeptide. Okay, so this line represents a polymer of amino acids. It represents amino acid after amino acid after amino acid after amino acid. Okay, and it's coiled round into the spring-like shape, like so. Okay, and this spring-like shape is what's known as an alpha helix. Now, what actually holds the polypeptide in this alpha helical shape? Well, let me show you this. So, to do this, what I'll do is I'll redraw this picture, but I will draw a amino acid actually out with its full structure at part of this. Okay, so I'm drawing the picture bigger now, and then we're going to have an amino acid that I'm actually going to draw out in full here. So here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Here's the R group. Now, I'm not bothered by what the R group is because this is a secondary protein structure and is therefore going to be formed just by the core structure of the polypeptide. Okay, it doesn't matter what the R group is here. Okay, so it's going to be formed by the groups of the peptide links. Okay, then we'll go back to representing the polypeptide just as a line, like so. It'll coil around like so. And now I'm going to draw another amino acid here. Okay, so once again, here is its amino group. Here's its alpha carbon with an R group here and a hydrogen coming off it. And then here is the carbonyl group of the amino acid. And then this structure will continue on. Okay, and you could continue it on. Right, now how are we going to hold this portion of the polypeptide here joined to this portion that's below it? Well, we're going to do it via hydrogen bonds here because we have the two ingredients to make a hydrogen bond here. We have a hydrogen attached to a nitrogen atom. Nitrogen has a much greater electronegativity, and that's going to result in hydrogen having a partial positive charge here. Okay. Then we've got an oxygen atom here, which is attached to a carbon atom. That's going to result in that oxygen atom gaining a negative charge, or a partial negative charge at least. And that oxygen is also going to have two lone pairs of electrons. We only need one of those, so I'll just show one of them. And then what's going to happen is this partial positive charged hydrogen is going to form a nice interaction with that lone pair of electrons that is on that partially negatively charged oxygen there. And this interaction is known as a hydrogen bond. Okay, and it's a very strong form of intermolecular bonding. So the polypeptide is held in this alpha helical shape via these hydrogen bonds that form between amino acids that are running parallel above and below each other, basically. Okay, so that's how alpha helices form. Now, let's talk then about helix-turn-helix motifs within proteins. So, basically, the name tells you exactly what you're going to find here. You're going to find a helix, followed by a turn, followed by another helix. Okay, so let's say we have the polypeptide starting here. Here's the amino terminus of the polypeptide. Now, I'm going to use a special little bit of nomenclature here. I'm going to put 2 lines like so, and then we'll continue the polypeptide on. Now, what does this mean? This means that there could be loads of stuff in between this point here and this point here, loads of other interesting domains. Okay, and now we're going to go into the helix-turn-helix domain, or the helix-turn-helix motif. Okay, so you're firstly going to have your first alpha helical structure. So let me show this here. Okay, so it's working its way up like so. Okay, and then it's going to turn around. Okay, so it's going to turn around like so. And that's the turn, and then it's going into the second alpha helix here. Okay, and then again, it will come off this alpha helix. I'll put this piece of nomenclature again, which means you could have lots of interesting domains between here and here, and then you'll have the carboxylic acid terminus. Okay, so we have these 2 alpha helices. Here, this one is in blue here, and this is our second alpha helix. Then we've got the turn portion, which is here in vivid purple. Okay, and then we've got the first alpha helix here in orange. Okay, so this is the helix-turn-helix motif, made up of a helix, followed by a turn, followed by a helix. Right, now the second alpha helix here is the important one. This is what's known as the recognition helix, and this is the one that's going to bind to the DNA. Okay, so depending upon the exact amino acids that you have within this recognition helix, it will bind to a set sequence of organic bases within the DNA. Okay, so it will bind to a specific recognition sequence, basically, and that will be determined by the specific sequence of amino acids here. Okay, so let me now show you how it's going to bind to the DNA. So let me draw my DNA here. So I'll start by drawing the first strand here, and then let's put the second strand here. Okay, so quite clearly we have the minor groove here, and then the major groove here. And of course, this recognition helix is going to bind in the major groove of the DNA. So if I draw this here, here is the recognition helix binding in the major groove of the DNA. Like so. Then we've got this turn, and then we go into the first alpha helix, which isn't going to bind to the DNA. And then of course we've got our gap, where you could have all sorts of other domains. Then you've got your amino terminus, then you've got your gap, where you could have all sorts of other domains, and then the carboxylic acid terminus here. Okay, so let's colour in these alpha helices. So this one is in blue. This is the recognition helix binding to the recognition sequence. Here is our turn in purple, and here is our first alpha helix in orange here. Okay, and this is just dangling down the side, basically. Okay, so a huge number of different transcription factors contain helix-turn-helix motifs. Okay, now, the other thing I should mention is about the dimerization of helix-turn-helix motifs. Okay, so often proteins that have helix-turn-helix motifs in, often they dimerize. Okay, and therefore you will have a dimer of these proteins containing helix-turn-helix motifs binding to the DNA, and I just want to mention how this works. Okay, right, so let's draw a fresh piece of DNA here. Okay, so here is one strand of DNA, and I'll have to draw it a little bit longer now because we want two of these things to be binding here. Okay, then here is the other strand, like so. Okay, right, so we'll have our first helix-turn-helix domain binding here, and to make it simpler I'm just going to draw the recognition helix as a blue line, and then I'll draw the first alpha helix as an orange line, like so. And then the other one is going to bind down here, and it's going to bind in completely the opposite way. So if we turn it upside down, we'll draw another one of these here. Okay, so here's the recognition helix, and here is the first helix in orange. That's how the other one is going to bind. So if we think about what the recognition sequences that you're going to need are, we're going to need this one to have another recognition sequence that's going in the opposite direction down here. So let's explore that in a bit more detail. So let's just make up a recognition sequence for this. Let's say that the recognition sequence for this recognition helix to bind is an adenine followed by a thymine followed by a cytosine, and therefore the complementary organic bases will be thymine, adenine, guanine. Okay, so here's one of the strands of the DNA, and here's the other strand of the DNA. Okay, now remember the two strands of DNA are running in opposite directions. So let's say this is the 3' one, and this is the 5' one, and that means, this is the 3' end of this strand, this is the 5' end of this strand, and therefore this strand will be running in the opposite direction. This will be the 5' end of this strand, and this will be the 3' end of this strand. Okay, so if we say that this strand here, and I'll colour it in just to make it utterly clear. Okay, let's say this strand in purple here, or pink rather, is equivalent to this strand here. Okay, and not in orange because we've used orange, but in green, this other strand here is this strand here. Okay, so this recognition helix here required an adenine followed by a thymine followed by a cytosine, and then on the other strand, which is going in the opposite direction here, okay, so let's just label them both up here, it required a thymine followed by an adenine followed by a guanine. This one down here is binding in the exact opposite direction, okay, so it's going to require the relationship to have reversed basically, but it's going to require the same sequence of organic base pairs here, okay, so let's think about this then. So we have our 5' strand is now the pink one, okay, and the 3' strand is now the green one. So effectively, the two strands have swapped places. Okay, so in the case of binding in this direction, we needed the strand that was coming from the 3' end to have adenine, thymine, and then cytosine, and then we needed the 5' strand to then have thymine followed by adenine followed by guanine. Okay, so if we use that same logic now going the other way, we need the 5' strand to have the thymine followed by adenine followed by guanine. Okay, so let's put those in. It's quite difficult, but we're going to have the thymine followed by the adenine followed by the guanine here. Okay, then on the 3' strand we need the other way round. We need adenine followed by thymine followed by guanine, so we need ATC there. Okay, so if we now turn it back up the right way, what we just have to do then is write those the right way up. Okay, so we need a thymine there, an adenine there, a guanine there, and then we need an adenine there, a thymine there, and a cytosine there. And this will be seen by this helix-turn-helix motif as being equivalent to the way this would have been seen by this first one. Okay, right. Okay, so that's how two helix-turn-helix motifs combined at separated distances along the DNA. So you'll have a few organic bases in between these. And now, of course, I've said that the transcription factors will have other domains other than just the helix-turn-helix motif, and these other domains will then be involved in binding these two transcription factors together. Okay, so this is what you need on the level of the DNA for two of these helix-turn-helix motifs to actually bind to the DNA. But then what we haven't shown in this picture is how the two transcription factors in the first place are binding together. Well, it doesn't involve the helix-turn-helix motif. It involves other motifs that will be in these sections which we haven't elaborated, and those will be specific to the actual transcription factor. Okay, right. Now, many transcription factors dimerize. This is a running principle for many transcription factors. Okay, right. So we're now going to talk about a special type of helix-turn-helix motif that you find in many transcription factors, which is known as a homeodomain. Okay, so this is a special structure that many transcription factors contain, which is contains a helix-turn-helix motif, but is a little bit more fancy than that. Okay, so it's worthy of note in its own right, basically. Okay, so homeodomains then. They have this helix-turn-helix motif in, however they have an additional alpha helix as well. Okay, so basically what they consist of is, here is their amino terminus. Okay, and again you could have in other domains before the homeodomain, and now we're going to start the homeodomain. Okay, so then you have your first alpha helix, which is one that's not actually going to be involved in the helix-turn-helix motif. Okay, you'll then have a large linker between this and your second alpha helix. Okay, and this one now is going to be involved in the helix-turn-helix motif. This is then going to go into the helix-turn-helix motif, like so, and this helix will go in front of this one here. Well, actually behind this one here. I'm viewing the front as being, sorry, yes, it's going to go into the page away from us. So imagine this alpha helix is in the plane of the piece of paper. This one is going to go behind it. Okay, right. And then, of course, you'll finish with the carboxylic acid terminus, but you might have other domains before the carboxylic acid terminus. Okay, so closest to us then is this orange alpha helix, which is the first alpha helix. Okay, so that one's closer to us. Then you have this linker that's in between the first alpha helix and then the second alpha helix, which I'll have in blue here. And then the second alpha helix is the first alpha helix of the helix-turn-helix motif. Here's the turn in that helix-turn-helix motif. I'll just override that blue in purple here. And then you have the second alpha helix of the helix-turn-helix motif in yellow here. And this is going to go behind the one that's coloured in orange there. Okay, right. So that then is a homeodomain. Now, the second alpha helix of the helix-turn-helix motif is once again going to be the recognition helix. It's the one that's going to bind to the major groove of the DNA. Okay, so let's show this. So once again, let's draw our DNA here. Okay, like so. Then we have our major groove here. I haven't really got enough space, but never mind. And then what we're going to have is this recognition helix sitting in the major groove here, like so. Then you'll have your turn, and you'll go into your second alpha helix in blue here. Okay, then the linker, and then the first alpha helix will come out in front of the third one there. Okay, so let's try and colour this in to try and get the message. In fact, I should have just done the colouring in first place. In fact, I think I'll start another picture. Ignore that one. Let's draw a better picture here. So, here is the DNA once again. Here's the minor groove. And then we've got the major groove as well. Right, okay, so what's going to happen then is we're going to have, I'll draw the first alpha helix again first. Okay, so this one is the one that's not going to be involved in the helix-turn-helix motif. You've then got the linker, then you've got the second alpha helix, the first of the helix-turn-helix motif, and then you've got the yellow one, which is the important one, the recognition helix, which is then actually slotted into the major groove. So I'll draw this larger to make it even more obvious. So there is the recognition helix sitting in the major groove, and then here is the C terminus that we've got over here. Okay, like so. And then this is the amino group over here. Okay, so that's how homeodomains bind to the major groove of DNA. Okay, right, so we'll call it there for this video, and we'll continue our discussion of domains which you can find within transcription factors in the next video.